So hello, everyone, and thank you for joining us for a Fireside chat as part of Mars Impact Health. I'm Janet Rossant, and I'm President and Scientific Director of the Gairdner Foundation. Gairdner celebrates scientific excellence through the annual Canada Gairdner Awards. These 7 awards go to scientists from around the world for their impactful biomedical and global health research. Gairdner also aims to convene leaders through our various programming and to inspire the next generation of innovators. To date, over 400 awards have been presented to laureates worldwide. 96 of those have gone on to receive the Nobel Prize. To learn more about Gairdner and our events, visit gairdner.org. And today I'm very happy to be joined by 2 of our 2022 Canada Gairdner International Award laureates. Dr. Katalin Karikó is Senior Vice President, RNA Protein Replacement Therapies at BioNTech, and an adjunct professor at the University of Pennsylvania. And Dr. Pieter Cullis, who's professor in the Department of Biochemistry and Molecular Biology, University of British Columbia. Along with Dr. Drew Weissman, close collaborator of Dr. Karikó, they were awarded for their pioneering work developing nucleoside-modified messenger RNA and lipid nanoparticle drug delivery, the foundational technologies for the highly effective COVID-19 messenger RNA vaccines. So thank you both very much for joining me today, and I'm looking forward to our discussion. So here we are. Amazing work from both of you and Drew Weissman, and many other people in this whole era of vaccines against the pandemic. And of course, the world was incredibly impressed by how fast it was possible to develop new vaccines for COVID-19, and in particular, the messenger RNA vaccines within less than a year, one went from the discovery of the sequence of the virus to having vaccines. So it sounded like it all happened overnight. And of course, you two know that it wasn't overnight at all, that there's a long process of discovery and setbacks, roadworks along the way, many different paths that led to the final outcome. So I'd like to hear a little bit about your story from the beginning of the fundamental research to the actual application. And I'll start with you, Katie. So you really were the person who was convinced that messenger RNA, which is one of the intermediates in how we translate genetic information into proteins, how messenger RNA itself could become a therapeutic. How did you start in that? And what was the most important thing that allowed messenger RNA to move forward to become a vaccine?
Indeed, it was not overnight. It was decades of overnight hard work, which led to, as me, Drew Weissman, and Peter was doing this. And I have to say that, thousands of other scientists, because the science is, is a group activity, and there is not I. It was we, and even those people who, came well before us, and they are not with us anymore. So we learned from them through decades. And for me, it was even, I am from Hungary originally. And when I started actually as an undergraduate, I started to work in a lipid team. And then we did the liposome and we delivered plasmids. And in 78, I started my graduate course and getting my PhD. And then I moved to the RNA laboratory. At that time, of course, I didn't know that the RNA and the lipid will be so closely associated. But working in the RNA laboratories, we tried to develop an antiviral compound, which was a short RNA, and we synthesized. And during that time, I learned how to work with RNA because it activated an RNase. I had to make template and other things. So manipulating the RNA, labeling, and working with different enzymes, it was important. And later on, I moving to the United States. I followed up in this RNA work. Actually, we did different modified nucleosides. Actually, we incorporated to the small molecule. And then from 1989-90, I started to write grants about mRNA therapeutic application. I never wanted to make any vaccine, no cancer vaccine, no infectious disease related vaccine. I thought that mRNA could be translated in every cell almost, because translation is ongoing. And then if we just deliver an RNA which codes for a therapeutic protein, transiently we can treat some disease. And these diseases, acute disease like aches and pains. And I was not thinking about genetic disease. I was just thinking about healing of a wound or something like that. And so I thought that for this one, you don't need to change the genome, because in the 90s, it was when 1990 started the Human Genome Project, and different genes were identified responsible for certain diseases. That was the trend, to introduce genes into the patient and try to fix their disease in that way. But I did not plan to do such things. I was focusing on just treating acute diseases and the mRNA coded for therapeutic proteins.
So that's what you were working on, but you didn't have an easy task, right? Grants were hard to get. You had to fight to make people believe that this was going to work. So tell us a bit about that.
Yes, indeed. This RNA was developed in, how we say it, in the shadow of the DNA therapy, because everybody was focusing on the DNA. It is permanent, and everybody considered, which was true, the RNA had just problem. It was, it was first of all, very small amount of protein could be produced because it's so quickly degraded. And that little amount of protein, everybody considered that it would be not enough to treat any kind of disease. In addition, as we learned with the work with Drew Weissman, this RNA was extremely inflammatory in human immune cells. And so the RNA just had a problem, too little, and it degrades and it's inflammatory. So if you see in the 90s, publications are coming out like once a year, and then you have never seen those people publishing again, because they just did find something and then left the field because they couldn't get maybe funding, or they had run into other problems. But I was lucky to have always at least one person who felt sorry for me, or who believed the RNA, and he provided at least the salary so I could keep going and doing. And constantly the RNA get better. And as Pete would say, in addition, parallel, the improvement of the delivery system. So it was first with lipofectin, which was good in cultured cells, but it was toxic in animals. And so, but parallel, it was developed better delivery system, and then RNA improved in a way that more protein could be produced from it. And that's we work, with the caps, different structural elements, purification, and other contributed to that.
Yeah. So it's things start to come together, and you start to see the lipids and the RNA coming together. So let's go back to beginnings of Peter's story then. So here's somebody who's worked on lipids for a very long time, Peter, is that fair to say? And how lipids are formed in membranes. So fundamental research. At what point did you start seeing that what you were doing as fundamental research was going to have, or could have, applications? Because in fact, you've been a kind of serial entrepreneur all the way through this. Pretty early on, you were trying to move this into a commercial applications. But tell us a bit about that transition from the lab to the commercial applications, and backwards and forwards, which has been really your life.
Yeah, it does go way back. When I got my PhD in physics in 1972, and I knew one thing at that point, which was I didn't want to go on to a career in physics. So I was quite definite about that. And the problems in outside physics looked a lot more interesting than the problems in physics. What was the bottom line. And particularly problems in the life sciences. And so there's a professor in the physics department who introduced we had a couple of people talking about membranes. I thought that sounds interesting. And so I applied for a postdoc to go to Oxford. This is an MRC postdoc, a Canadian MRC. It was MRC at that time, not CIHR. And to go to Oxford, and so I got that, which was kind of a big surprise. It was certainly a really big surprise when I I went into the biochemistry department in Oxford and walked past the animal room, which was right by the entrance to the department. So nothing like a physics department at all. So that was a real reality check. So for a year or two, I it was really quite interesting, knowing nothing about biochemistry and didn't know what a protein was or nucleic acid, and eventually started to catch on, and I knew quite a bit about NMR, so I did NMR on biological membranes and particularly on lipids, and I found it quite fascinating that here was this very essential part of biology, and the biological membranes are absolutely vital. And we knew so little about them. There's thousands of lipids in a biological membrane, and we don't have a clue what they do. Not now even, for the bulk of them. And so there was two aspects I cottoned onto. One was lipids are supposed to be in a bilayer in a membrane, and yet when you pull them out of you extract them from a membrane and then put them in water, they adopt these non-bilayer structures. So what's going on there? Why are they there? And the other was lipid asymmetry. The lipid composition on one side of a membrane is different than the other. And so, I wondered whether or not we could get ion gradients to distribute lipids across membranes and see what the functional consequences might be. So that's a start, and from that, a lot of you were asking about the applications, etc., really came from 20 years of trying to understand those problems. But at one point, we found that we could make these small liposomal systems and use a pH gradient to load drugs into them. And this was some of our lipid asymmetry studies. So this in the mid 80s, I was trying to keep a very good team that I had together, had at UBC. And I thought, okay, well, if other people can start companies, I'll start a company. And so we started a company to deliver cancer drugs more accurately to where they're needed in the body. Then, of course, you run into the realities of companies. The mid-90s, the CEO came to me and he said, look, putting these old cancer drugs into liposomes is all very well, but I can't raise money on that. I need to be doing gene therapy, which was coming into vogue at the time. And so this meant we had to encapsulate DNA, RNA into these lipid nanoparticles. Well, that was a huge problem. Because the cationic lipids, in order to encapsulate something in a hydrophobic or lipid nanoparticle, you needed to have something that had a positive charge, lipid with a positive charge that associate with a negative charge on the RNA or DNA. And those molecules are really toxic in vivo, and we'd learned the hard way with our studies on with liposomal formulations of cancer drugs that toxicity will kill a drug every time. It's so we knew we couldn't use these available cationic lipids. Well, as luck would have it, we'd synthesized what we termed an ionizable cationic lipid as part of our studies on lipid asymmetry, because we could flip that across the membrane in response to pH gradients. And so we thought, okay, we'll try this because it had the property that at low pH, it was positively charged, whereas at neutral pH, it wasn't. And so we could perhaps encapsulate at the low pH, encapsulate the nucleic acid at low pH, and then bring the pH up. And then we asked the question, well, at that point, we were working with antisense oligonucleotides, so were they retained? It turned out they were, we now had a system that was much less toxic, but where we managed to very efficiently encapsulate the nucleic acid. So to fast forward on that, we then started working. This is in the 2000s with a company in Boston called Alnylam. Want to deliver the small interfering RNA to the liver, to silence the gene in the liver. And 7 years, that was a remarkable collaboration, actually. We really did manage to get a system where we could silence the gene in the liver with a therapeutic index around about 1,000. So in other words, we could give 1,000 times higher dose of the medicine of the lipid nanoparticle plus the siRNA before we saw any toxic side effects. So we took that into the clinic, and it's fantastic results coming out in 2018. But at the same time, about 2012, when it went into the clinic, we couldn't do any more work on it, because when something goes into the clinic, that's kind of it for a scientist. You just have to, hope for the best. We thought, okay, well, maybe we can deliver messenger RNA to the liver, which also turned out to be possible, to my great surprise. And see proteins being expressed. So instead of silencing a protein in the liver, we were now expressing a protein in the liver. And this is where Katie and Drew Weissman came into the picture, contacting us to say, well, gee, it's all very well to be producing proteins in the liver, but how about if you go in intramuscularly and try and see whether or not they have properties as vaccines? That turned out to work brilliantly. But I just went through that a bit because it points out the very zigzag, nonlinear, whatever way you want to state it, way that you get to something that obviously has had some important applications. But this was not a linear path. I mean, we weren't going for vaccines at all, but we developed developed systems that ended up being very, very useful.
So we are here, two of us. We didn't want to make vaccine and turn out. This is how science works. You are working on something and you don't know what it will be useful for.
Yeah, Katie and I give lectures together sometimes, and it's the audience must be wondering, well, how did this happen? You weren't going in that direction, but you're going in completely different directions, and but anyways, as I said before, that's just the way that science operates. You certainly have to be aware of, it points out the importance of collaboration in the end. I mean, if you're not collaborating with somebody that maybe has another viewpoint, another application, then you're going to miss out on a lot of opportunity, that's for sure.
No, I think it's a great story, and as you say, you have to come together. But both of you took different routes, but all the way along, you were developing new techniques and new models. So the opportunity was waiting, and you took it. Both of you took it. So, Katie, how did you get in touch with BioNTech and sort of move partly from academia into industry? How did that play out?
I started to move away from academia actually in 2006 when we together with Drew Weissman, based on our discovery, we established a company. So I established only one company. Pete established several other ones, but we did one, and this was for nucleoside modified RNA, and we proposed that erythropoietin coding mRNA could increase the hematocrit, and it would be good to treat patients who had anemia. Okay. And we submitted a grant for the STTR grant to the government, and we got first the $100,000, then the $900,000. So total is about $1 million small business grant. And we did demonstrated that the erythropoietin coding mRNA could increase the hematocrit in mice. So that was it. But why we are not talking about RNA-Rx, why it is Moderna and BioNTech. We could not get our patent from the university. It was given to somebody else. So then to go to the industry, so I went to BioNTech. I accidentally learned about existence of this company. That company had no website and nobody knew about it. And the reason I went there, really, I could have gone to Moderna as well, that BioNTech had a clinical trial mRNA, and we are in 2013. And so I was thinking Moderna did not have a clinical trial, didn't have a GMP's quality RNA. So I thought it will take for decades. We didn't know that it will be within a decade it happened, just I assumed it. And so I thought that if BioNTech has already an RNA in clinical trial, they know how to make RNA. And then I wanted to make sure that, you know, I said that I will come here if I can work in nucleoside modified RNA. And Ugur Zain said that, yeah, I can work on it. And that's how I got there. And then it's, you know, it was a very difficult move for me because my family is in the United States. So I commuted to Germany. And many people said that, you know, I'm going to the wrong direction because usually from Europe, they are coming to, to the US. But, you know, I went to Germany and started there. And it was like, I was already 58 years old. So I retired from the university and made this move to go there and started to work to introduce the nucleoside modified RNA at BioNTech. And so the company at that point, when I went there, it was in a campus, you know, and it was different buildings, and it was just less than 100 people work probably, and maybe 70 people were on a production site, which was a 20 years old production site, which was purchased, which was already producing plasmids and cells, and this was a GMP facility. But the scientist group was much smaller than today. You know, it is 2,000 or 3,000 people there, but it was in 2013 was a much smaller company. And so I was very delighted, actually. I mentioned because finally it was not important how many papers and what impact factor and whatnot was important, but we have to make a product which is helping somebody, which is good and functional, and everybody work together. It was different than in academia when, okay, we just mentioned we work together, but it's more competitive, you know, when you are in your field and somebody is close to your field, and then you have to publish, you have to be the first or last author, and you know, and there is more competition on that way also. And here, nobody's counted whose name is what. We just have to have a product, and that was very uplifting for me.
So that's interesting that you would say that. I think that's an interesting message for people to hear, because obviously, especially students, they often think, oh no, going into industry, I'm going to be constrained, I'm going to lose the ability to really do what I want to do. But what you're saying is, you know, in some ways, because you're not having to publish in the right place, you don't have to get grants, if you have a goal and you have the means, you can really start to achieve that goal. So.
But you are right on the essence that well, those people, because you won't do what you want really, maybe, you know, because if you go to a company, you have to do what those who established the company. If it is a smaller one, you have to do that. And so you have to make your own company if you want to do exactly what your final goal is. But yeah.
So I mean, but that's the way things have to move, right? There is this freedom of discovery, but if you have a discovery and you see the potential, it's interesting so both of you have taken that and said, I want to take it all the way through. I want to see it translated into something that helps people. And not everybody wants to do that. Certainly there are plenty of people in academia who will just be quite happy to try and get their next paper in Nature and Science. But it's a different mindset. Peter, you've not only set up your own companies, but of course you've been very much involved in the sort of life sciences community and life science commercialization community, particularly in BC. You were one of the founders of what is now Admare as a commercialization center. How important is it really to develop those kind of activities so that the discoveries that we do make can be translated in Canada? I'm going to go Canadian now, Katie. How can we see that we can do better at making sure that discoveries can be translated into impact in Canada?
It's a really important question. I spent, I've done 3 not-for-profits that have really been aimed at, this is the CDRD, Center for Drug Research and Development, which is now Admare. I also started something called the Personalized Medicine Initiative, which is still in a to push in that direction in terms of introducing precision medicine. And the nanomedicines or nanomedicines innovation network that is across Canada, initiative aimed at bringing the nanomedicines efforts across Canada into have some commercial reality. And I think it's this is something where we do need to double down, I think, in Canada to really get that mindset in place that perhaps it's a good idea to the discoveries that are made in the universities. Perhaps it's a good idea to really facilitate the potential for those discoveries being the basis of companies in Canada. But as you're aware, 40% of our science, technology, engineering, math graduates go south, basically taking jobs in the US. So that, if you say it costs us $200,000 to educate those people in their PhD, there's about 5,000 graduates each year. Means 2,000 are going south. So you can do the math. We're subsidizing, American industry to a remarkable extent. And people don't go south because they don't want to stay in Canada. They go south because that's where the jobs are. And so it's extraordinarily important for us to, as Canadians, to say, look, this is a real priority, and universities can do, I think, much better in encouraging these kinds of endeavors. Facilitating the movement of intellectual property from the university to startup companies, as well as encouraging finding ways to fund things in the early stages, which is where most things die, most initiatives die. So for me, it's a real driver. Now, the other part of this, and Katie alluded to it, is that it's a heck of a lot of fun. If you're a part of a team that is trying to do something, and you're in a small company, it's one of the most enjoyable things you can ever do. It's a very that sense of we're going to do something big together, and everybody pitching in. It's a really it can be a very rewarding experience. And it is very different from academia, where in order to make your name, you have to have some individual academic research, whatever achievements. Whereas in a company, particularly a startup company, we're all together in it, and so the group success is the individual success. So that's one of the messages that I want. That for me, it's a very big, very good way of getting it's a very exciting career to get into. So from the audience point of view, I'd encourage them to think about this. And it isn't something that is scary, or there's the profit motive is there for sure, but the discovery and actually doing something that could be a benefit is a bigger driver, that's for sure.
Well, there's no question, of course, that the two of you, Andrew, and all the other people who've been involved in vaccine development in the last couple of years have had an enormous success, and it must be extremely pleasing to know that what you have done is really, saved lives. That's an incredible thing to have achieved. So congratulations to both of you. And it's interesting, the Gairdner Awards, I see that every year we give awards to some of the best researchers in biomedical science. And, the ones that are most effective, the ones that are most exciting are those where it's been possible to go from discovery to something that really impacts on humans. So, this year, Stu Orkin is one of the other awardees. And there you're seeing gene therapy and CRISPR, gene editing, finally going from, very rapidly, really, from, again, discovery to something that could really and does impact on people with genetic diseases. So, but he didn't go into the company himself, but he everybody who worked with him spun out and ended up in those companies. So I think it's this combination of going backwards and forwards between academia and companies is something that should be absolutely embedded in the way we think about doing, particularly biomedical science these days. Katie, I'm going to turn to something that I know is close to your heart as well, and that's women and women in science. If you look back on your career, how do you feel? Do you feel that you were given a fair shot as a woman? Do you feel that there were barriers in the way? And do you think things are improving for women in science?
I mean, for me, it was important to stay in science when I have my daughter, who happened to be a 2 times Olympic champion, 5 times world champion, a rowing legend in the United States. Susan Francia, she was rowing in a Canadian boat also, I mean. So she was, just 4 months old. I could leave her in a very high quality and affordable childcare. That was important, that the government, if the government is listening right now, they can do something for the women, affordable, high quality. That's very important. So I could work easily and stay in science. And for me, I did not experience that because I am being a woman that I was, handled differently. Maybe sometimes, because I have an accent, I was 18 years old and I couldn't speak English. So, heavy accent. And then, sometimes they assume that, they asked me, when I gave like the first talk after, about the nucleoside modified RNA, who is my supervisor, they assumed that that woman had to have a supervisor. But again, who asked me was another woman. So I wouldn't say that it was a man handling differently. And this was the same way that who demoted me was another woman, from my position. So I wouldn't say that. And who supported me, like Elliot Barnett, and who hired me at Penn, and David Langer, who made sure that, neurosurgery, 17 years I can work there. So they were supporting me financially and they were men. So my thinking is that somebody is a good leader, whether he's a man or woman is fine. And any leader who is, a little bit compromised. Or the other thing is what thinking is that if it at home, you have the more equality. So your husband is helping you and appreciate you have a dream and you want to work on it, then sooner or later in the workplace, it will be the same. So that's, we have to work on it, but definitely, is for the woman is somewhat more difficult, because if one family, then still she had to be pregnant. And so it is not easy, but if she gets help, and they had a good partner, then together they can manage it. And so I can, and also I am an alien or an immigrant here. So that's also, I went through all of these, what many scientists also in Canada is going through, that they are coming from another country, and immediately that process, immediately adjusting to the changed environment, and immediately learn how to swim. You are just gonna drop in the water, and sometimes, against the flow, you have to learn and quickly adjust. And that's also very helpful in science, because you always like a new situation and how to solve it. So I just can encourage all of those women and immigrant scientists, who I am myself, is that just, keep doing the good job, focus on what they can do, and don't worry too much about and comparing themselves to the others. And just what they can do, they have to focus on that. And then they will be good at, and they have to enjoy, of course, the work.
Well, I think that's great messaging to women, underrepresented groups in science. If you have a passion and you're really dedicated, then keep going. That's also what I always tell people. However, the numbers tell us that women are still not represented in the scientific community at the levels that one would expect. And if you're certainly in academia, as you go higher and higher up academia, the numbers of women get fewer and fewer. So we still have to, I think, look carefully at what are the barriers. And you mentioned one of them, and it's so silly. It's childcare, affordable childcare, accessible, easy childcare. It's amazing how that is still actually a big block for many women and young families in science. So something we need to work on. So anyway, it's good to see strong women like you making a difference, and obviously hoping to be a role model for the future. So let's get back to just finishing off now. Let's return to what we started, which was the discoveries and the applications, the messenger RNA vaccines and the technologies involved. And you both said, well, you know, when you started on lipid nanoparticles, when you started on messenger RNA, you weren't thinking about making vaccines. So what else can we use? Messenger RNA, lipid nanoparticle delivery systems, well, messenger RNA and delivery systems for what do you see the future in the next few years for this kind of technology? It seems that there's now a lot of interest. So, you know, your companies, BioNTech and Moderna, they were fairly well funded, but now I think there's going to be a lot of activity in this area. So what do you see as the sort of next top hits for messenger RNA vaccines?
I mean, you just mentioned the genome editing. I have to say that the first real success is, again, using mRNA with the Intellia, which is another company in Boston area that used the messenger RNA coding for Cas9 and the guide RNA, and they were successfully could permanently stop making that toxic protein in the amyloidosis patient. And of course, what you from the news now that daily is coming out that the new vaccine trial initiated, and Moderna has HIV vaccine, which we don't have anything. It was a Epstein-Barr viral vaccine, again, two different trial is ongoing and started, and you could also see that some of the vaccines which we already have, but it was not affordable, because that we did not mention about that LNP mRNA. It is that this is cheap, and so it makes many other medicine affordable. And so you could see that against shingles vaccination is already this year will start both Moderna, BioNTech, Pfizer. And so replacing something that is like €800, the present vaccine in Europe. So it is, for shingles, for example. So you can see also not just viral disease, but intracellular bacterial disease like TB. You can see also that parasites like malaria vaccine, and of course, in clinical trial, many other things, even for tick, and against toxins and other things you can have. And the big section of the industry is growing in pharmaceuticals was the antibodies, which is very expensive again, because the protein had to be purified. So those delivering the mRNA coding for the antibody, the patient can make the antibody itself. And so it is a passive vaccination, and also for infectious disease as well as for cancer treatment is already ongoing. And I am protein replacement. So what am I doing? Protein replacement, and BioNTech together with Sanofi, we initiated injection of messenger RNA coding for cytokines, and then it will make a cold tumor hot, and then help the immune system to recognize, and circulated cell will recognize the remotely located tumor. And that's ongoing clinical trial. So that was one I participated at.
It's just extraordinarily exciting to see the potential here. And what about lipid nanoparticles beyond nucleic acids, Peter? So obviously they play an incredibly important role for the nucleic acids, but you can put all sorts of things in them as well. So what's big in your side of the fence?
Well, I think that the application to mRNA, siRNA is probably the biggest thing that we're, I do have some other things going on, but it's just amazing. I mean, what Katie just recited, the number of applications that are coming, that are being exploited by various companies. I refer to this really as the third generation of pharmaceuticals. First generation small molecule drugs, second generation biologics, and then the third generation is this, you can call them gene therapies, but anyway, using nucleic acids as therapeutics. And we got touched the surface of this. It's truly incredible. I mean, there's all the vaccine applications that Katie just went through, and that it's a huge number. Then there's chronic disease. For example, Drew Weissman and some of his associates just published something using a CAR T-cell therapy to treat the fibrosis of the heart. In other words, to treat heart failure. This is one example. And then you have all the rare diseases Tay-Sachs, Fabry's, whatever. And so if a child is born now and is not making a particular protein that is essential, I mean, this which happens fairly often. I mean, 20% of the people in of people in hospital, children in hospital, are suffering from these kinds of conditions. 20% of all diseases, I think, actually are rare diseases. Anyway, you now have this possibility for saying, okay, well, they're not making such a protein, let's make the mRNA. And within a very short time, it takes you a day to formulate, within a very short time, you have a very focused, personalized therapeutic that is affordable. So it's completely revolutionary. We're moving into an era of personalized therapeutics, and I think people should start to realize that up to this point we've been dealing with a one-size-fits-all approach to medicine, and that so we all get prescribed one thing, but anyway, it's as if these things are suited to all of us, which of course they're not. And the fourth leading cause of death in North America is an adverse reaction to a prescription drug. 90% of adverse reactions aren't reported, so it's likely much higher than that. We do need a much more personalized approach, and I think this is the potential now for analyzing somebody, saying, okay, you're missing this or that, and then getting a very personalized LNP mRNA to produce whatever protein it is, and doing that in a time period that's real, like in real time, a month or 2 months, is going to shape the future of medicine in a really fundamental way. Now, just with regard to lipid nanoparticles, right now we're good at the liver, and obviously the vaccines are working quite well, even though they were designed for expression of proteins in the liver. So there's probably some optimization that we can do there. But getting to bone marrow, getting to the lung, getting to these other tissues, getting particularly to the brain, all of which is going to happen, and we're seeing early signs of that. With every stage of that, you have incredible new therapeutics that are going to result. So it's a revolution in medicine, and is something that is well, obviously the pharmaceutical or the pharmaceutical industry. The industry that's concerned with developing new medicines is aware of that. You're seeing billions of dollars go into this area. As new companies pretty much every day that are being set up. Venture capitalists are really crowding this space, and it's an amazing time. And I'm trying to say, okay, in Canada, we need to double down on the opportunity that we have. We were caught a little bit with our pants down by the pandemic, having our pharmaceutical industry has been decimated over the years. Now is a chance to correct that as we move into this new era. It's being estimated that up to half of new medicines in 10 years' time may very well be based on this kind of approach, the nucleic acid approaches. We need to be part of that story.
I think that's a great message. The concept of personalized therapeutics is really going to change the way that we think about medicines in the future. Canada needs to be there. And the other thing, the message that you also both said, is that these kind of therapeutics can be not expensive. They can be affordable. They can also be things that could be applicable into global health issues. So we could really see this going from the individuals to the major problems that we have worldwide. So it's a very exciting time. It's been wonderful to talk to both of you, and congratulations again on the fantastic work that you've done and that you continue to do. And we all look forward to a great future in personalized therapeutics. Thank you.
Thank you.
Thanks, Janet.